So why do we need to change the way we look at soil, particularly on mine site or construction sites, Paul?
Well, the main thing that we have to do is look at the way that soil is constructed. Mainly when we go on a mine site, they've stripped away the topsoil, stockpiled it for 6 months to 3 years, and it's turned into a dead, inert dirt. Effectively, all the nutrients have been washed out, the microbiology is completely gone, and we need to reconstruct it once it's put back out. Likewise with the subsoils, they're usually pretty nasty and full of constraints, whether they be high sodium levels or compaction or dispersive soils. So we actually need to get into rebuilding the soil structure, soil health, and along with that, it comes with the soil biology.
Revegetation doesn't usually fail because we lack inputs. It fails because we don't create the biological conditions that make those inputs work.
That's exactly right. So in the chemical, conventional way of dealing with soils is we can throw a lot of water-soluble nutrients at it. We can go and adjust the pH because the pH is too low or the pH is too high. We can throw gypsum and things at it, and that can bring the sodium levels down, but it lacks biology. In our biological systems, what we do is we put in beneficial microbes as an inoculum. We feed the microbes so that they then look after the plants. And because of that, we reconstruct the soils. And it's not the lack of input that causes soils to fail or revegetation to fail. It's the lack of biology and conditioning the soils so that the microbes can live in it.
So what are soil microbes and why are they essential?
Basically, soil microbes consist mainly of bacteria and fungi, and the ones in the soil that we're interested in are the beneficial microbes. So the beneficial fungi like mycorrhizal fungi and trichodermas, and then the beneficial bacteria that create healthy rhizospheres and also help in nutrient cycling, so that we can actually then use those microbes to grow around a seed that's developing, create a rhizosphere that's healthy, and within that rhizosphere, the microorganisms will buffer out the constraints that are on the outside. So, for instance, if we're in a acidic, aluminium-dominated soil, most plants don't like growing in that. What happens is the microbes create this envelope or rhizosphere around the root system that buffers out those constraints like the aluminium. And so we can do a pH test on the soil that might be pH 4.5, and if we go into the rhizosphere, those microbes create it so that it's maybe pH 6, which is conducive to plant growth. So that the actual roots are not actually touching the environment, but they're protected by the microbes that are around it.
So microbes aren't just an add-on, they drive the mechanism that makes the soil function.
That's exactly right. This is all about creating a living, dynamic soil, and the microorganisms within that soil will drive the system. So that's why we get such good revegetation, good seed strike, good resilience of the plants, because the microbes are creating this really healthy, dynamic living system.
So what's the quickest, most efficient way to build microbes in soil?
The best way of doing that is inoculating the soil with a suite of beneficial microbes. So in our biological system, we use 24 different specific microbes that create a rhizosphere and condition the soil for the system. We then have to feed those microbes. So we use control-release fertilisers that are made predominantly of natural recycled mineral ores, and those feed the microbes, not the plant. The fertiliser feeds the microbes feed the plant. rather than in a water-soluble system where you're applying fertiliser that will feed directly the plants. And the third thing that we need to do is create a habitat. So as the roots develop, the microbes create a rhizosphere around those roots, and that makes the roots grow better, particularly into constrained soils, compacted soils, high sodic soils and so on. It allows the roots to develop, and the more roots, the more habitat there is for the microorganisms. We add biochar that stimulates microbial activity and act as a catalyst to form biofilms. And these microorganisms then grow carbon within the soil. And so effectively what we're doing is we're buffering out the horrible parts of the soil, the constraints, and creating an environment that's more beneficial to the system.
Microbes need seed, feed and shelter. Give them all three and they'll switch the soil back on.
That's exactly right. So as we said, we put the microbes in, we feed them with the controlled release fertilisers, and we give them a habitat to live in. That means then the seeds are growing into a much more conducive environment than into the constrained soils that we generally see in rehab situations.
You mentioned biochar. What is biochar?
Yeah, so biochar is a very interesting compound that is derived from pyrolysing or burning biomass under low oxygen conditions. Biochar has really come about in agriculture and environmental work in the last 20 years, but it was originally developed as a terra preta 2,000 or 3,000 years ago by the Aztecs and Incas in the South America. What it does is it acts as a habitat for the microbes to live in to begin with. So when we inoculate microbes into the soil, they're in a hostile environment. So they'll go and live inside the biochar and then grow out from the biochar, forming biofilms. And these biofilms are then the fixed carbon. The second part about them is biochar is very stable, or recalcitrant carbon, we call it. And the stable carbon will be there for tens, if not hundreds of years. And so this contributes towards keeping carbon within the soil up. And the importance of the carbon in the soil is that it keeps the EH level, or the redox potential, at a high level. And we'll probably discuss redox in a minute. The third thing is that biochar acts as a catalyst for the microbes to grow on. And so, as I was saying, the biochar, from the biochar, the biomass of the microbes grow and we get these biofilms forming. Those biofilms help in stabilising soil structure and in creating soil pores. And in the pores, we have oxygen, because this is an aerobic system. And when it rains, water can infiltrate and be stored in those pores. So effectively, the carbon that grows off the biochar acts as a water sponge within the system.
So in the biogrowth system, biochar is used as both a microbial habitat and a structural amendment that reinforces long-term soil health.
That's exactly right. And I mean, we've done over 20 years worth of research into using biochars in really hostile environments. The main part about them is that they act as this habitat for the microbes to establish themselves. And then once they're established, they help the root development of the plant that we're trying to grow there.
What goes wrong when microbes are overlooked?
This is the biggest question in soil biology. Up until recently, soils have been treated as a chemical and physical world, and as a consequence, we've basically made our soils dependent on junk food fertilisers, water-soluble fertilisers that we have to apply that wash down past the root systems. The problem with the water-soluble ones is that they're highly inefficient, so they've got poor nutrient use efficiency. That means that when, say for instance, you add a nitrogen product like urea, about 30% of it washes straight past the roots. 30% of it volatilizes, comes out as nitrous oxide. And only 30% is available for the plant. As a consequence, we've got about a 30% nutrient use efficiency. If we treat all of our soils like this and our plants like that, we've got a very inefficient system. And that's why we go onto a lot of mine sites and rehabilitation sites, and we see good strike, good growth for the first 2 or 3 or 4 weeks where there's a lot of nitrogen around. Then they run out of steam, and those plants then are susceptible to the constraints that are put on them. That means that you get short-term cover, but patchy establishment and weak soil structure. The biggest problem with the conventional system, particularly with applying a lot of water-soluble nitrogen, is we burn the carbon in the soil. Most soils have some organic carbon in there. If you apply nitrogen, for every nitrogen atom that's metabolized in the soil, you need 10 carbons to do that. That's the biological process. And what happens is that you burn your soil carbon. Once you burn the soil carbon, the soil structure starts falling apart. And once the soil structure falls apart, then you get dispersive soils, or you get hard setting soils. Going the other way, when you add biology to the system and you can complement the chemical and physical world with biology, we grow microbes into the system. We use less nitrogen because we've got nitrogen-fixing microorganisms in our mix. We grow carbon within the soil rather than using it. The carbon then builds structure into the soil, builds those air pockets which allow water infiltration, and that builds structure back into the soil. As a consequence, we get better revegetation, so plants grow better. They have water held up in the root system in those air and water pockets. And the third thing is that they then stabilise the soil structure so you don't get erosion occurring.
So without biology, soils become dead zones, plants hit the wall, and projects can fail.
That's exactly right, and this is what we see time and time again. As a soil scientist and a researcher, we spent most of our time fixing up problems that had been caused by inefficient programs. Now with the biogrowth system, we're able to go in and fix up those problems. So at a commercial level, we're able to do programs that build the soil biology, build up the structure of the system, increase the water use efficiency and infiltration, so then the plants are more resilient, and with the microbes in that rhizosphere creating a good environment, the plants are more resilient and more self-sustaining.
What's unique about the microbial suite in the EnviroStraw biogrowth technologies?
Yeah, so basically these are what we call bioenergetic microbes. We've selected a suite of 24 different strains of microbes, so bacteria and fungi, and they're selected for specific reasons. The main ones are in creating a habitat around the root zone that is protecting it or buffering it from the constraints within the soil, so high acidity, high alkalinity, high sodium, high aluminium. These all might be toxic to the plant. The microbes create this rhizosphere to protect it. So that's the first thing. Second thing is that we have mycorrhizal fungi, and these attach to the roots and grow out, and they can grow up to 1 metre away from the plant and pull nutrients and water from a distance, so the plants aren't constrained by fertiliser being at distance. It can be brought into the plant. That means you don't have to put as much fertiliser out. The third thing is that we have nitrogen-fixing microorganisms in our suite. And these things like Azospirillum or Azotobacter, they live in the root zone and basically fix nitrogen for the plants. So they pull nitrogen out of the atmosphere, and our atmosphere is about 70% nitrogen. So effectively they can pull nitrogen into the plant through the nitrogen-fixing bacteria for free. And so this is really a big part of the self-sustainability of the biogrowth system, is that you don't have to keep reapplying nitrogen. Your plants don't run out of steam, nitrogen steam, after 2 or 3 weeks because the water-soluble nitrogen's disappeared. The nitrogen-fixing microorganisms produce amino acids and nutrients that can be taken up by the plant. The other important thing about the nitrogen-fixing microbes is they produce plant growth hormones, and these help in stimulating root growth so that we get bigger root growth. That creates a bigger habitat for the microorganisms to live in. And then the third thing is that they produce plant growth hormones that force the plant up so that we get more biomass formed. And so we get good ground cover, better strike, and ultimately better biomass above ground creates shade, and then you don't get so much evaporation of water. So it's a water conservation thing. So the next set of microorganisms that are important are the ones that sequester carbon. Photosynthate from the plants are secreted through the root systems, and up to 50% of a plant's sugars are secreted into the root zone. Why would they do that? It's very expensive to produce sugars from photosynthesis. It's pulling carbon dioxide out of the atmosphere and it's secreting it into the root zone. That is to stimulate beneficial microbes in the root zone. These microbes then take the carbon and deposit it in the soil. And so from that we start again to build the soil carbon sponge within the system, and that helps with water holding capacity of the soil. And more importantly, in exchange for those sugars, the microorganisms bring back nutrients or water to the plant. Finally, the other set of microorganisms we have are stubble digesting microbes. These are ones that break down cellulose within the system, and a lot of the soils that we're dealing with are hydrophobic. That means that water can't penetrate, so we get rainfall. It basically runs straight off down the batter and it's either lost or it causes erosion. With the stubble digesting microbes, they can eat the glycocalyx, which is this like plastic skin that forms on the surface of soils. These microbes use that as a food source, an energy source, and by eating the glycocalyx in the soil, they allow water to penetrate. The water then penetrates, goes down into the carbon stores, the carbon sponge in the soil, and holds on to that moisture so that when things dry out, our plants are more resilient because they've got more water and last longer.
So biogenic microbes are a diverse microbial team that can do what single strains can't, build a dynamic living rhizosphere that drives revegetation outcomes.
That's exactly right. So the key to it is these diverse species of microorganisms and the different strains that we use are specifically selected for environmental outcomes. We're going into really harsh environments, these disturbed soils, and we've selected microbes that can grow in those soils and create a living dynamic system out of an inert dead dirt.
So what are bioenergetic microbes and how do they enhance the biogrowth system?
Yeah. Bioenergetic microbes have been specially selected. They're microbes that can grow in really constrained soils in really harsh conditions, and the main thing that they are involved in is in microbial signalling. So they're discussing with the plant what the plant needs in the system. So the plant will pay with photosynthate and the bioenergetic microbes will go out and find those nutrients and pull them in. The second part is that the bioenergetic microbes are really designed to sequester soil carbon. So they'll take the exudates that come out of the root system and lay them down as biofilms in the soil and in developing root structure, in developing soil structure, pore structure, and so on. And the mycorrhizal fungi, as part of the bioenergetic microbes, they'll go and explore the environment for nutrients at a distance from the root, which is not usually available for that plant. And the final thing is that these bioenergetic microbes are really good at breaking down things like non-wetting soils. So the biggest problem we have in both agriculture and in rehabilitation is that the soils become non-wetting, and the microbes can go out and eat the glycocalyx, which is like a waxy substance that forms on the soil surface. These bioenergetic microbes like to eat that glycocalyx as a nutrient. That allows water to flow into the system, and so we get better water infiltration, and that water then gets stored in the water sponge.
So bioenergetic microbes switch on the soil-plant conversation, building a dynamic rhizosphere that drives resilience from the ground up.
That's exactly right. The most important thing is developing a dynamic living soil, and that builds resilience into the plants that we're trying to grow when we're doing revegetation work.
What role does mycorrhiza fungi play?
Probably one of the most important set of microbes in the soil system, particularly in regenerative revegetation, are the mycorrhizal fungi. They're basically symbionts, which means that they form a symbiotic relationship with the plant root. So the fungi attaches to the plant root and then grows out and extends way beyond the reach of the roots. And that gives access to nutrients and water at a distance, which aren't usually available to the plants. When they grow out, the hyphae that grow out are very fragile and can be easily broken. So these hyphae from the mycorrhiza give out a compound called glomalin, which is like concrete carbon, and that protects the hyphae so that the conduit of pulling nutrients and water into the plant isn't broken. The importance of glomalin is it's a very recalcitrant carbon, meaning it's long-lived in the soil. It'll be there for 10, if not 100 years from now. And so the importance from mycorrhizal fungi, one is to pull nutrients into the plant, water into the plant, but two is to lay down this glomalin concrete carbon that helps form soil structure, tensile strength, pore spaces, and ultimately build structure into the soil. And on disturbed soil sites, this is generally the thing that we find most absent, the mycorrhizal fungi.
So mycorrhiza are the plant's underground internet, extending roots, sharing carbon, holding soils together.
That's exactly right. So the largest living organism on land are mycorrhizal fungi. There are forests in Europe and the United States where they've measured these over 1,000 hectares, and they're single living organisms that attach to all of the trees within the forest and pull nutrients in. And along there, they have signalling aspects where they can tell plant systems if there's issues forming. They can help pull water into the system, they pull nutrients into the system, and most importantly, they stabilise the soil structure